Now, you guys know me, I'm a little bit of a tech nerd. I love anything to do with technology and especially things that help move the world forward. And so, what we're going to talk about today is technology and one specific technology. Now, you guys watched the video talking about how our knowledge of cells came to be. And there was a piece of technology in there that was crucial to the development of what we now know as our modern cell theory. And that was the microscope. And so we're going to talk about the microscope in its current form and how that's helping change our perspective of the world as it is now. So, when we talk about the microscope, the first thing that's probably worth talking about is the magnifying glass. Now, a very crude microscope, this has just one lens and everything we see is illuminated by light. The one that we're probably most familiar with when we say microscope, this is probably the image that comes to your mind, is the compound light microscope. Now, this has two or more lenses and we still use visible light to illuminate our subject specimen. And so in this one, we've got a ocular lens and then our magnification lens, and we've got these focus arrays that move our thing up and down so that we could get it in focus. And then normally we have some kind of light either shooting up through it or down onto it. And we get images like this. Here we see the borders of the cell, we see the nucleus in the middle, and generally it requires some kind of staining to help define what we're seeing. And so these are like our traditional compound light microscopes, but where I want to focus our energy today is on electron microscopes. Now, these work a little bit differently. Instead of sending visible light through, we send electrons to illuminate our specimen. And so we're going to talk about two types of electron microscopes today. We're going to talk about transmission electron microscopes, TEM, and we're going to talk about scanning electron microscopes, or SEM. Now, both of them look similar to this. This is a pretty generalized picture, but they're large on top of a big desk with a computer beside. And while we're not going to talk about how they each work, we're going to talk about the image that these each produce. And so let's start with the transmission electron microscope. The transmission electron microscope, when we send electrons, they are passed through the specimen. That's why we call it transmission. They pass through the specimen to produce a 2D image. Now, these are some of the most detailed images we've seen up to this point. They allow us to see the structures inside because it's passing through. That's our transmission electron microscopes. And then the next type was our scanning electron microscopes. And instead of passing electrons through our specimen, a beam of electrons create a 3D image. Now, to me, scanning electron microscopes are the coolest because you can see the type of images that they produce. We see these cells in incredible 3D detail where we see the surface features and the texture and the structure of it. And so they often appear fuzzy, and I just think their images that they're produced are so cool. Not to say that scanning is better than transmission. In fact, transmission is better for some things than scanning. But for example, this photo here is actually a photo of the novel coronavirus, what we're experiencing right now. This is taken with a scanning electron microscope, and we're able to see structures and for scientists, hopefully identify binding sites so that we can create a vaccine for this thing. So the real advantage of these electron microscopes is what it allows us to see. Where light microscopes aided and helped us see things beyond our human eye, the electron microscopes, transmission and scanning electron microscopes, allow us to see things almost down to the atom level, which allows us to understand things in a brand new way, ways that we've never seen before. And so I want to talk about 4 technologies that have emerged out of this and that are helping us understand the world around us better. And what we're going to do is I'm going to briefly summarize them, and then you're going to choose 2, and you're going to go ahead and research them. There are links to get you started and some really simple questions that you're going to answer about the 2 that you've chosen. So let's talk about the 4 tech that we're going to look at. The first one is gene sequencing. Now, our human genome, all the DNA that makes up who we are, DNA is composed of 4 bases: A, T, G, and C. And in these combinations, we create all these long lists of these specific sequences. And these sequences tell us who we are and give cells information on what they're supposed to do. And so they set out to sequence the genes to understand what is the order for all of them and what does it all mean. So one of the things you can research is, why is this such a big deal? And you'll see those questions in there. So gene sequencing, step number one. Technology number two is cell culturing. We now have the ability to grow cells in a lab, independent of our body. Now, we know that life needs to come from life, and so we are able to take cells that exist and grow more of them in the lab. This is really important in things like in the medical field, as far as like skin grafts and organ grafts. There's lots of research happening in that, but it's also important in understanding things like cancer and how cancerous cells grow and multiply so we can someday find a cure. So cell culturing is technology number two. Genetic engineering is the third one, and to me this one has a lot of intrigue because there is some ethical things that go along with it. Essentially, genetic engineering says we're going to take out part of a gene and we're going to splice in something new. You may have heard of this when people are genetically modifying plants so that they last longer, or vegetables, but this is also being talked about in the context of humans. What if you could change your baby's eye color before they were born? Genetic engineering. Got some good things going and definitely some question marks, another thing you could be researching. And then the final technology is stem cell research, and this one's fascinating as well because before cells differentiate and become their specialized version with specialized tasks, they start off as stem cells that essentially have infinite possibilities of what they could become. And so stem cell research is looking at, how do we harness that? How do we harness the cells that haven't differentiated and become something specialized? And maybe we could tell it what we want it to specialize in. And so these are four technologies that I want you to take a look at. You're going to choose two of them. I've given you some starter resources on the slides below for each of those, and then some short questions that you're going to answer. What is that technology and why does it matter? So go ahead, give that a try.